Hello all, my name is Miles and this is my IPM presentation. Unfortunately, my computer does not support any type of recording whatsoever, so I'm forced to do it on my phone. Um, for our group project, we focused on precocine 2. As you see to your right, you have the chemical makeup of precocine 2. Precocine 2 as an IGR or insect growth regulator affects insect growth stages, also affects behavior in certain species of insects. In others, it sterilizes and or kills them in the adult stages. Precocin 2 is derived from plants in the Ageratum genus. Ageratum Itisi, Conzoides, and Houstonianum are the three main plants Precocin 2 is derived from. Ageratum iltisi is native to Ecuador, extracted and refined to make the IGR Precocin 2. It is used as a ground cover and is actually a weed depending on the location. Ageratum conizoides, also called goatweed, is native to Brazil, and everywhere except Brazil it is considered invasive. Ageratum houstonianum, known as the blue mink or blue weed, is native to Mexico and used as a bedding animal. There's about 60 different species of Ageratum. There's actually 4 native to the US. Apparently, these are the 3 most popular for making precocine 2. Professor Bruce D. Hemlock from the University of Vermont, his studies on precocine 2 started in 1979. There was another professor who also did their research around that time, but another one of my group members grabbed him. He first did his research on Oncopelotus fasciatus, or the milkweed bug. It affected the insect's endocrine system, or Capora alata. The Capora alata secretes juvenile hormones. He found in his research that precocine 2 was most effective when the alata was active, about 1 to 7 days after hatching. It effectively shut down the endocrine system, and it also caused an issue in the egg yolk formation called vitellogenesis. There's a picture of Dr. Hemlock, a picture of a Capora alata, or the insect endocrine system, and a, what was that? One more time. Milkweed bug. I think this is the cockroach Capora alata. I couldn't find one this detailed for the milkweed bug. The next insect he did his research on was the locust migratoria, or migratory locust. It also affected the insect's endocrine system, or Capora alata. Instead of neutralizing or stopping the progression of its maturation, the locust, it actually collapsed the Capora alata altogether, and it led to the death of the locust. I guess apparently the hemocytes, or white blood cells, attacked the affected tissue, and this research was able to classify it as a cytotoxin when it came to the migratory locust. He also did research on a brown-banded cockroach, or Supella longipalpa. It affected the cockroach's ability to secrete sex pheromones, and it disrupted the mating cycle. It also affected the Capora alata, which it did in most. I think that's the main mode of attack, is attacking the endocrine system. Our next 2 are M.J. Hayes and J.H. Oliver out of Georgia Southern College. They're part of the biology department, and they conducted their studies in 1981. Their research in 1981 was featured in the Journal of Parasitology. They did their research on the American dog tick, or the Dermocenter variabilis. It's native to the Rocky Mountains, and it vectored something called tick paralysis. That sounds fun. The insecticide used to control the American dog tick at the time was petroleum-based and killed just as many beneficials, if not more, than the tick. Studies on precocine 2, it sterilized the females and affected maturation in both male and female. In the areas where they applied with precocine 2, they saw a 50% drop in the American dog tick, And they had noticed, which was peculiar, that there was a dramatic drop in males. So that was just something that stood out. Another um, person was Dr. Raj Kamal Srivastava from Cambridge University. He conducted his trials in 1992, and his findings were published by the Cambridge Press in 2009. He conducted his studies on Aphis crackivora, or cowpea aphid. Similar to results of the maturation in other insects, the endocrine system was affected, leading to issues in instar formation and not being able to mature to a full aphid. Something that he did notice, that wing growth was not affected by precocine 2. That was just something that stood out in the research. And then I do believe this is our last researcher. This is Shohei Sakuda from the University of Tokyo. His research for precocine 2 was funded by the University of Korea out of Seoul. And instead of insects, he used his on fungi. Gibberella zeae, or the Fusarium gramineum, it affects a lot of cereal grain crops such as wheat and barley. As you can see I don't know if you can see that on the picture on the left, the top is a white color and the bottom is a normal green. It vectors in as a pathogen Fusarium head blight. Precocine 2, when applied to the fields, inhibited the fungi's production of trichothiocin. This is a mycotoxin leading to Fusarium head blight. Immediate results were noticed, but more research was needed. I think this is a student, Shohei, because I can't find any pictures or anything else other than these articles he wrote on fungus. So why is precocine 2 not as so popular with all the positive results we see? For one, its effect is very expensive. The 16-ounce bottle to the left, I don't know if you can see that, cost about $63. And I can see some of the larger scale farms and producers having an issue with that price, with that amount, especially when you can buy certain insecticides at a bulk price for a lower amount. So until they figure out how to manufacture this at a cheaper rate, I can still see it being effective as an insect growth regulator, but it won't take off until you find a cheaper way to produce it. What else do we have? And that concludes my presentation. I want to thank you guys for listening. And again, I do apologize for everything.